Good morning or afternoon, depending on where you are, and welcome to today's lecture. I'm happy to see most of you again. I think that we already met in chemical biology a month ago. And today's lecture will be structured into two major parts. First, we will explore research methodologies and associated statistical analyses used across different types of experiments, studies, and surveys. And second, we will apply these ideas specifically to vaccine development, one of the most critical areas where robust research design directly impacts global health outcomes. And by the end of the session, you should be able to differentiate between experimental, observational, and survey-based studies, understand which statistical tests are appropriate for each, and recognize how these designs and analyses apply in real-world settings, particularly in vaccine trials and approvals. So now we can begin. So when we talk about research methodology, we are referring to strategy, design, and logical structure that frames a research project. You can think of it as building a house. You wouldn't just start by laying bricks randomly. You actually need a blueprint, a plan, and materials selected for specific reasons. Similarly, in research, methodology provides the blueprint to systematically ask questions, gather information, and draw valid conclusions. A weak methodology undermines a study's credibility, no matter how exciting the results might seem. And this is why it's important always to have a really strong methodology before you start collecting your results. Research designs fall into three broad so-called families. We have experimental studies, observational studies, and survey-based research. Each design type has strengths, weaknesses, and fits certain kinds of questions better than others. Choosing the wrong design is like using a hammer when you actually need a screwdriver. You might get some result, but it won't be reliable or appropriate. And this is why it's important beforehand to determine your research questions from which you can determine which type of study to conduct, particularly for these research questions, and then start developing your methods. Otherwise, if your study is not appropriate, then your results will not be valid either. And now we can begin with experimental studies, which are the most rigorous form of research. Here, the researcher manipulates one or more variables to observe effects. Typically, researchers assign participants randomly to different groups, such as treatment versus placebo. And the gold standard here is randomized controlled trials, known as RCTs. So some key features of randomized controlled trials are, firstly, randomization, as also said in the name, which reduces bias. This is very important because we don't want healthier or more motivated people to accidentally end up more often in one group than the other group. And randomization ensures that known and, more importantly, unknown factors that could influence the outcome are distributed equally between groups. Here I'm going to give you an example of vaccination already. And imagine we're testing a new flu vaccine. If younger and healthier participants all end up in the vaccine group by accident, and older, failure participants end up in the placebo group, then the results would unfairly favor the vaccine. So randomization prevents this imbalance or bias. Secondly, we have blinding in randomized controlled trials, which would be either single or double blinding, and it prevents expectations from influencing outcomes. Blinding means that participants, researchers, or both do not know which group participants are assigned to. We have two major types of blinding. Firstly, we have single blind studies where only the participants don't know if they're assigned to placebo or the treatment. And we have double-blind studies, which are the most rigorous form of randomized controlled trials. And there, neither the participants nor the researchers know the group assignments, whether placebo or treatment is assigned to a patient. Blinding is important because expectations can influence outcomes. And you might have heard of a phenomenon known as the placebo effect or observer bias. So an example here would be if a participant knows that they received the vaccine, they might believe that they are protected or report fewer symptoms, even if the vaccine had no real effect. Or on the other hand, if a researcher knows that someone received a placebo, they might even unconsciously be more attentive to symptoms in that person. And this is why double blinding protects the integrity of the data by minimizing these psychological and behavioral biases. And we know that controlled conditions like this maximize internal validity. And in a randomized controlled trial, we standardize as much as possible. So we standardize how interventions are delivered, how the outcomes are measured, and how participants are monitored. And we do this because the more tightly we control external factors, the more confident we can be that any differences in outcomes are truly due to the intervention, for example, a vaccine or a certain drug, and not due to some external confounders. And this is also called maximizing internal validity, meaning the study is valid inside its own experimental system. I will again give a vaccine example. If you have a vaccine trial and one group is allowed to take additional antiviral medication and the other group isn't, then any difference in outcomes between the two groups could be due to the medication and not the vaccine. And this is why we have strict protocols that ensure that both groups are treated identically except for the vaccine exposure. So the only difference between the groups will be the vaccine. One group will be receiving the vaccine and the other one will be receiving a placebo. And this allows us to actually identify the effect of the vaccine, not from another factor from the outside. And if we turn to statistical analyses, we have different kinds for experimental studies. Firstly, we have t-tests, and these are used when you want to compare the means or averages of two groups to see if they're statistically different from each other. As an example here, if you have a clinical trial testing a new COVID-19 vaccine, and one group receives the vaccine and the other one receives the placebo, then after, let's say, 4 weeks, you measure the average antibody levels in each group. And you would use an independent sample t-test to compare the two means between the group that received the vaccine and the group that received the placebo. And you could be able to answer the question if the average antibody level significantly higher in the vaccinated group compared to the placebo group. So this is the benefit of a t-test. However, we have to note that t-tests have certain assumptions. So firstly, the data has to be approximately normally distributed, and the two groups should have roughly equal variances, which is the spread of the scores. And if these assumptions are not met, which can happen if you have a small sample size, then we can use another test, which is known as the Mann-Whitney U test, and it's a non-parametric test. So t-tests would answer the question, are these two groups meaningfully different in their outcomes? However, now you may ask what happens if we have more than two groups. Then you have the analysis of variance, known as ANOVA analysis. An example here would be that you are testing 3 different doses of a new flu vaccine. You have a low dose, a medium dose, and a high dose. So you have 3 groups instead of 2. And you want to know if the average immune response differs between the 3 groups. And here, if you would run multiple t-tests between every pair of these groups, it would increase the risk of type 1 error, which is finding a difference by chance. But instead, if you use the ANOVA, it compares all group means simultaneously under one overall test. So this is the benefit of using an ANOVA instead of t-tests when you have more than 2 groups. And if the ANOVA shows a significant result, it tells you that at least one group differs from the others, but it doesn't tell you which group. And then you perform the so-called post hoc tests to find exactly where the differences are in your data. And here again, you have to assume that there's a normal distribution within each group, equal variances across groups, and you must have independent observations. And ANOVA is essential when you're comparing multiple interventions, doses, or experimental conditions at once. Further, we have regression analysis, which helps us adjust for other variables called covariates that might affect our outcome. So as you know, in real-world experiments, not every participant is identical. We have factors such as age, pre-existing medical conditions, gender, socioeconomic status, and these all can influence results. So if you have a new randomized controlled trial and you're testing a vaccine, then older participants may naturally have weaker immune responses. And if you want to see the pure effect of the vaccine, then you must adjust for age. And we have a linear regression model, which allows you to estimate the effect of the vaccine on antibody levels while simultaneously accounting for age, gender, or any other factor that you want to adjust for. And you would use a linear regression if your outcome is continuous. For example, predicting someone's antibody level after vaccination based on their age and health status. And if your outcome would be binary, such as just looking at infected or not infected, then you would use a logistic regression instead of linear. So linear, you can remember if we're talking about any antibody level between, let's say, 250, then 500, then 1020, it's a continuous range. And that's why it's linear. But logistic would be used for binary if we just have infected or not infected. So there are just two categories that are possible. And this regression is about isolating
Paula, can I ask a quick question?
Yes, sure.
So when you say logistic, are you using logarithms?
Yes, so when you run a logistic regression in a statistical software such as Stata, it will give you the logs, and this is why it's also known as logistic regression.
And you can actually mix logarithms with variables that aren't in logarithmic form too, right?
Yes, you can, yeah.
Yeah, that's really interesting, very powerful. Not many people actually know that.
Yeah.
Okay, go ahead.
Yeah. So regression is about isolating the true effect of your experimental treatment while you're controlling for, let's say, background noise from any participant differences among the whole population that you're studying. And lastly, we have Kaplan-Meier curves and log rank tests. This is not used so often, I would say, in research, but it's actually very useful because here you aren't just looking at if the event actually happened, but you're asking when did it happen. And this is why it's called a time to event analysis or survival analysis. So an example I would give here is if we're testing a vaccine and we want to know how long does it take until participants get infected after the vaccination. And participants would be followed over time, and some clearly get infected and some don't. And if you have this Kaplan-Meier curve, then you would see a proportion of participants remaining event-free, meaning infection-free over time. And the curve only drops when somebody experiences the event, so they get infected. And a flat line means that there are no new infections. And then, so basically we want a flat line in this case in the Kaplan-Meier curve. And you can compare the curves of the vaccine and placebo groups, so you can overlap them. And you can see, and hopefully with the vaccine, you will see a more flat line or only a few drops. But with the placebo, most likely you're going to see more drops of more people getting infected. And the log rank test tells you whether the difference between curves is statistically significant. And as you see, experimental studies are powerful because they can establish causality, so cause and effect relationships, and they don't just give us an association or a correlation. So we're actually able to state in our study at the end of the results that we have a cause and effect relationship. So we can say whether the vaccine is actually effective, to what extent, and be able to draw these conclusions. Further, we move to observational studies. These happen when randomization is not possible. And sometimes it can be due to ethical reasons, cost, or feasibility. And then researchers turn to these observational studies. Here, as of course the name already mentions, the researcher observes natural exposures and outcomes without interfering. And there are different types of observational studies. So we actually have three main types. Firstly, we have cohort studies. So you start with a group exposed to a risk factor and a group not exposed, and you're going to follow them over time. An example here would be following vaccinated and unvaccinated individuals to observe infection rates. Then we have case control studies, where you start with people who already have an outcome, known as cases, and you compare them to people without a certain outcome. So they are the controls. And you look backward for exposures that caused them, these cases, to have these outcomes. An example here is that among people who developed a rare vaccine side effect, we look at exposures that they shared in the past. And then we have cross-sectional studies where you take a snapshot at a single point in time. So you're, for example, surveying a population to measure current rates of vaccination or attitudes towards vaccines. And of course, we have some statistics here as well. Firstly, we have chi-square tests, which are used to compare categorical data. So data that falls into certain categories like yes or no, or vaccinated or unvaccinated. And we use chi-square tests to answer questions like, is there an association between two categorical variables? So if we're running a study on COVID-19 vaccination and infection rates, then we collect data from a vaccinated group where, let's say we had 100 people and 10 of them were infected. And we had an unvaccinated group also with 100 people, but we had 30 infected people instead. So as you see, both the vaccination status and the infection status are categorical variables. And we want to know if being vaccinated is associated with a lower infection rate. So just, of course, looking at these numbers, we already see, because we have the same sample size, so we see that less people got infected if they were vaccinated. But normally we would set up a 2x2 contingency table, which is just a square with like just a table 2x2, basically. And you use a chi-square test to see if the differences between the groups are bigger than what would be expected by chance. And this contingency table, the rows represent two classifications of one variable. So you have, for example, positive, negative, and yes and no. So yes intervention, no intervention. That's why it lines up as a 2x2. And some important things to think about the chi-square is it tells us if two variables are associated, but it doesn't tell us how strong that association is. And it works best when sample sizes are reasonably large. So with a small sample size, it wouldn't really work. And you should use chi-square when you're comparing proportions or frequencies between two or more categories. And then we come to logistic regression. So a small reminder from the previous, when we looked at experimental studies, we said that linear regression is for continuous outcomes, such as predicting antibody levels over a continuous spectrum. Whereas logistic regression is for binary outcomes such as yes, no, or having the event or having no event. And some examples in vaccine studies would be, did the participant get infected? Yes or no? And for example, did they experience an adverse event? Yes or no? So you might ask, how does logistic regression work? So instead of predicting a raw number, it predicts the probability of an event happening. And then it converts this probability into odds and models it using a log scale. This is why we say logistic. And what do we actually get from logistic regression? You might have heard of this term already, but you get odds ratios. And having an odds ratio of greater than 1 will mean that you have higher odds of the event happening. And odds ratios of less than 1 means that you have a lower odds of the event happening. And if we're interpreting this, then if the odds ratio for infection in vaccinated individuals is 0.2, then it means that vaccinated people have an 80% reduction in the odds of getting infected compared to unvaccinated individuals. And then you might ask, why would you use a logistic regression instead of just a chi-square test? Well, logistic regression allows you to adjust for confounding variables such as age, gender comorbidities, and things that could independently influence infection risk. And logistic regression not only tells us if there's an association, but it also quantifies the relationship while adjusting for other important factors. And lastly, we have Cox proportional hazards models. I think that in the research you hear of these a little less than you do, for example, the logistic regression. And these models help you when you have time to event data, such as the time until the antibody levels drop after receiving a vaccine. And it's not just, we're not interested in whether something happens, but also when it happens. So the same as with the experimental studies, we talked about the curves. So here it would be, for example, how long does it take after vaccination before somebody becomes infected? Or how long until antibody levels fall below protective thresholds? And here again, then you might ask, why not use just logistic regression? Well, logistic regression only cares about having the event or not having it, but it ignores the time. And Cox models incorporate both. So whether the event occurred and how much time actually passed before the event occurred. And the Cox regression works by modeling the hazard rate, which is essentially the risk of an event happening at a particular point in time, given that it hasn't happened yet. So here it gives us a hazard ratio. So if logistic regression gave us an odds ratio, then here it's a hazard ratio. And the hazard ratio of more than 1 means higher hazard or risk of the event happening. And the hazard ratio of lower than 1 means lower hazard or risk of the event happening. An example here would be that if the hazard ratio for infection after vaccination is 0.5, then it means that the vaccinated group's risk of infection at any given time point is half that of the unvaccinated group. And you would use the Cox models when you care about both the occurrence and the timing of the event. And remember that observational studies can show associations, but they struggle to prove causality, so cause and effect relationships, because of potential confounding variables that you are not controlling for as you would in an experimental study. And then we have surveys and questionnaires. So survey research is incredibly important, especially when we're trying to understand public opinion, knowledge, attitudes, or behavior. And surveys collect data efficiently from large groups. And I personally, my Parkinson's research is a lot survey-based because I look a lot about the knowledge and attitudes that people have towards certain treatments for Parkinson's disease, and also how they make shared decision-making with their physicians. And I'm able to do this in larger groups because I can send out the survey to thousands of patients. And even if a certain proportion of them actually respond to the survey, then I still have a large group. And key design elements of surveys and questionnaires are firstly the sampling method. So random samples are ideal. And convenient samples, for example, having students at one university, can introduce bias. So if you're conducting a survey and you want to survey students because you're looking at, let's say, their attitudes towards their online studies, you would rather take students from all over, let's say, even if you're just looking at the United States, you would take a bunch of universities from the United States instead of, let's just say, focusing on your university being Atlantic International University. And you also have to look at questionnaire design. So we must ensure that questions are clear, unbiased, and tested for validity. So measuring what you intend to measure, and of course, reliability, that you will have been consistent results. And here you will often hear a definition known as a validated survey or a validated questionnaire. So sometimes, especially in clinical research, you will use a survey that has been validated by somebody because you know that it's following a standard. For example, right now I'm running a survey study on shared decision-making in Parkinson's disease, and I am using the so-called shared decision-making questionnaire of 9 items, and this is SDMQ9, and it's already a validated survey. So this is making sure that my study is reliable and valid. And statistics for survey studies often involve firstly descriptive statistics. You will give percentages, means, and standard deviations. And of course, you're also going to explain describe actually your population, so who participated in your survey study. This is why you use percentages to say this many males, this many females, this many percentage was from this certain age group and from regions that they come from, so for demographic data. And of course, you're also going to have correlations. You're going to look at if two variables are related. For example, if higher education level is correlated with greater vaccine acceptance. I can give an example from a survey study I conducted and already published. It was on pharmacological treatments for Parkinson's disease. I found that higher education level was correlated with higher knowledge of more pharmacological treatments among patients with Parkinson's. And we also have factor analysis. So when you have many related survey questions and you want to understand underlying themes such as trust in science, and this is something that comes up in vaccines, especially after the COVID-19 pandemic, we wanted to survey people and see if they're actually trusting science in general if they were, let's say, hesitant to getting the COVID-19 vaccine. And what's important to note is that a poorly designed survey, so let's say having a not validated survey or not having thought of certain aspects of what exactly you're going to do and having a well-developed methods will just lead to garbage data, and it will happen even with a big sample size. So you need to really think through your methods, think through your survey questions, and have multiple reviewers who are going to read through it to say that this survey will be reliable and valid. Otherwise, it will give you garbage data. And statistical analysis with survey must fit the data and the question that you are asking. So you have major decision points. Firstly, what type of data did you collect? Was it categorical, ordinal, or continuous? So categorical data refers to labels or categories that have no inherent order. So each value belongs to a distinct group, but there's no ranking between them. So we have vaccination status. Are you vaccinated or not? You might have blood type, AB, or O. And for example, a type of virus, influenza, COVID-19, or RSV. Then ordinal data still uses categories, but now there is a meaningful order among them, even if the distance between categories isn't exactly measurable. But you would have, for example, a pain scale. So you had no pain, mild pain, moderate pain, or severe pain. Then education level, high school, bachelor's, master's, doctorate. And survey responses such as strongly disagree, disagree, neutral, agree, or strongly agree. So as you can see, we already have a special order among them, and we don't just have certain categories that don't have a rank. And then we have continuous data, as I already mentioned, which are numerical, and the numbers are meaningful and measurable. So you can perform arithmetic operations like addition, subtraction, and you can calculate means, standard deviations, and etc. And here examples would be antibody levels measured in units per milliliter, body temperature in degrees Celsius, blood pressure, or age. And you can also ask if the data is normally distributed. So you can also generate this using statistical software. I prefer to use Stata. And you can generate a histogram and see if the data doesn't follow the normal distribution, then you would need a non-parametric test. So if you're comparing means between two groups, you would typically use the t-test. But then if the data doesn't show to be normally distributed, you would use the non-parametric test known as Mann-Whitney U-test. And question is also how many groups do you have? So if you have two groups, you will typically use a test that compares the two groups. So as we saw also in the experimental studies, you have a t-test for continuous data, or then you would have a chi-square test like we just described in the observational studies for categorical data. And if you have three or more groups, then you will usually need the test such as ANOVA, as we discussed again in experimental studies, or an extended chi-square test. And you're also asking if your groups are independent or they are paired. So independent groups, participants in one group have no relationship to those in the other group. For example, you just have vaccinated people and you have unvaccinated people, and they are different. But paired groups would be that the same participants are measured twice, and for example, there could be a matched design. So we might measure antibody levels before and after vaccination in the same individuals, or we would match participants based on characteristics like age, gender in a case control study. So we're ending up with the same participants being measured twice in different conditions. And why does this matter is because when data is paired, then the two measurements are linked and they're not independent anymore. And you have to use a different statistical test. So you can no longer use the independent samples t-test as you did for independent groups. And you would use the so-called paired t-test. And an example for categorical data would be for independent groups, you would use a chi-square test, but for paired observations, you would use a mock NEMARS test, which again, I would say I have seen less in research because we are usually using independent groups. So here's an overview of what statistical tests you would choose. So if you have two independent groups and you are looking at the means, you're using a t-test. If you're comparing more than two groups, you're using an ANOVA. If you're comparing proportions, you're taking the chi-square test. And then you have the time to event outcomes for Kaplan-Meier survival analysis. So all of this might seem like a lot, and I myself have to cheat every time I do a study. I have a cheat sheet like this that says if you're looking at this, you have to use this statistical test because it's almost impossible to remember all of them. And sometimes another good way to identify which test to use is to look at other studies, somebody who published a study, and see what types of tests they used for their conditions. And from that, you can figure out what exactly you need. And one thing that you shouldn't be afraid of with statistics is to obviously make mistakes because it happens. You might choose the wrong test and you will realize it later, but you will go back, you will adjust your methods and you will rerun your analysis and you will learn from that mistake for the next time. For one of my survey studies, I had to rerun my analysis 3 times because I kept choosing the wrong statistical tests and I wasn't understanding why I'm not getting the results I was expecting. So this is something about statistics that you have to accept, that it is a hard discipline and it takes time and patience. And of course, you have more concepts that you need to understand in statistics. You might have heard of p-values. When you run a statistical test, you usually get a p-value. And this value tells you if the null hypothesis were true, so if there were actually no difference between the groups, how likely would it be to see a result this extreme just by a random chance? And if the p-value is small, it suggests that the result we observed would be very unlikely under the null hypothesis. So we might consider rejecting the null hypothesis. And here you have a common threshold that a p-value of under 0.05 is often considered statistically significant. And then you can reject your null hypothesis. Further, you have so-called confidence intervals. A confidence interval gives you a range of plausible values for your estimate. So we often talk about the 95% confidence interval, meaning that if you repeated your study 100 times, about 95 times, you will have the true value falling within the reported interval. So if you estimate vaccine efficacy at 80%, then the 95% confidence interval could have been from 70% to 88%. And you are pretty confident that the true efficacy will fall somewhere between this range, between the 70% and the 88%. So you know that in real life, it will never fall exactly at 80%. And a narrow confidence interval says that you have more precision, whereas a wide confidence interval means that there's more uncertainty. And if the confidence interval for a mean difference does not include 0, so it's either both of them are below or both are above, then you know that the result is statistically significant, even without looking at the p-value. And if the confidence interval for an odds ratio or hazard ratio does not include 1, so it's either going to be under 1 or above 1, then it's also statistically significant, even without you looking at the p-value. And confidence intervals show both the direction and the precision of your results. And of course, p-values and confidence intervals tell you whether an effect exists, but effect size tells you how big that effect actually is. And this is very important because a tiny effect can be statistically significant if the sample size is huge. But a tiny effect might not matter in real-world practice. So we have an example of a vaccine reducing infection risk by 2%. If we had a big study, it would be statistically significant and the p-value would be even under 0.01. But then the question is a 2% reduction enough to change public health policy? And the answer would be probably not. And types of effect size measures would be different in means. That would be for the t-tests. There's a so-called Cohen's d, which looks at standardized mean differences. Then you have the odds ratios for logistic regression and hazard ratios for time to event outcomes. And effect sizes tell you whether your finding is just statistically detectable or whether it's actually clinically or practically meaningful. And lastly, we have power analysis. So this might seem like not a big deal, but it is. And it's important that we talk about this before we conduct a study. So power is the probability that your study will detect a true effect if a true effect actually exists. And usually researchers aim for 80% power, meaning that you have an 80% chance of correctly rejecting the null hypothesis if the alternative is true. And power analysis is done before you start the study. Question is why? Well, firstly, we need to calculate how large your sample size needs to be. This is important for anything, for an experimental study, observational, also for survey studies. I conduct a power analysis to see how many participants I need from each group for me to have the 80% power. Otherwise, my study is, let's say, worthless. And in this case, you also avoid wasting time and resources. If you will calculate a power for a study that you've proposed and it will give you an unrealistic number of participants that, for example, you are sure you cannot get using survey data, if you're conducting a survey study, then you would just not do the study just because you know that it would be a waste of your time and resources because you would not reach the participant amount that you're required. And if we look at the vaccine research, then if you're testing a new vaccine and you expect it to reduce infections by 50%, then you have to do a power analysis on how many participants do you need to reliably detect this difference. And if you guess wrong or you use too small of a sample, then you might fail to detect the real important effect, even if the vaccine is working. And that would be called an underpowered study. And for this, we also have a statistical software called G-Power, and it's actually very easy to use because you input certain values and it gives you, you're saying which statistical test you're using. So if you're using a t-test to compare two groups, then you input that data and it will give you a power size that you need, so a sample size. So for example, 60 participants in one group, 60 in the other, or something like that. And now we move fully to vaccine research. So I did already give you examples of vaccine research when we were talking about certain statistical tests, but now we can apply this to vaccine development in more detail, which is a real-world high-stakes application of everything that we just covered. And as you know, vaccines are held to exceptionally high standards. And why is this? Well, because they are given to healthy individuals, and often millions of them, to prevent disease. So we are looking at preventing something before a person is actually already sick. And that's the difference between vaccines and medications. And the pipeline from the lab to pharmacy involves years of careful research, multiple study phases, and constant statistical scrutiny. So the major phases of vaccine development are, firstly, we have preclinical studies. You have probably heard of these. This is testing in cell cultures and animal models before moving on to humans. Then we have clinical trials. We have phase 1, which is testing safety in small groups of people. We have phase 2, which is testing safety and immune response in already larger groups of people. And we have phase 3, where we test efficacy and rare side effects in an even larger group of people. Then we have the regulatory review, which is a detailed independent analysis of the trial data that you conducted. And lastly, we have post-marketing surveillance, which is sometimes known as phase 4 studies, where you are doing long-term safety monitoring in the real-world population. So even though we call it phase 4 of a certain research study, this is already happening after a certain vaccine or medication is approved and it has gone to the market. And each of these phases uses different methodologies, and the stakes keep getting higher. So in the preclinical phase, researchers test candidate vaccines on cell cultures and animal models, such as mice or primates. And endpoints include safety, so looking if there are any toxic effects, and immunogenicity, so does the vaccine trigger antibodies or T-cell responses? And here you would use descriptive statistics mostly, so means and standard deviations. And of course, if you have small animal sample sizes, then you would require non-parametric tests. But the important point here is that if it's not safe and promising in animals, it will not move forward to humans. Then phase 1 moves into humans, which is typically fewer than 100 participants. And the main questions here are, firstly, is the vaccine safe at different doses? And are there any serious adverse events? So sometimes the design here is open label, meaning that everybody knows if they're what they're getting. So if they're getting the placebo, the vaccine, at what dose. And often we're not comparing here. So we're mostly focusing the statistics on adverse event rates. So how many participants experience side effects like fever, pain, or any allergic reactions. And we describe these rates, not just report percentages, but also provide confidence intervals to show uncertainty. So example would be 10% of participants reported fever, with a 95% confidence interval from 3% to 22%. This means that the true rate could realistically fall anywhere between the 3 and 22% based on the small sample size that we were using. And here you can see the confidence intervals are crucial because early studies usually have small sample sizes, which means that we have more uncertainty. And in these early trials, we don't usually do formal hypothesis testing, so we don't have any t-tests or p-values because the sample size is too small for meaningful inferential statistics, and we're mostly looking at descriptive statistics. So we are focusing on detecting safety signals if we have any unexpected or severe side effects, and we're asking if this vaccine is safe to move forward to bigger studies that include more human participants. And of course, we're watching for anything dangerous and not trying to prove statistical differences yet. Then phase 2 trials involve hundreds of participants. But now we are adding placebo controls or active comparator groups, meaning we compare the new vaccine either to a placebo or an existing vaccine. We measure immunogenicity, the immune response, usually by checking antibody titers after vaccination. For statistics, if we are comparing mean antibody levels between the groups, then as I said, we're using a t-test because we're looking at 2 different groups. Or if we are looking at different doses and more than 2 groups, then we're using the ANOVA test. To compare rates of side effects, like how many people report fever or muscle pain, we use the chi-square test because these are categorical outcomes. And if we want to adjust for participant differences, for example, if the vaccinated group is a little older or has more comorbidities, then we would use the logistic regression to adjust for those outside factors. And phase 3 is known as the critical proof stage with thousands or tens of thousands of participants, which may sound a lot. But this is also important because the participants are randomized to vaccine or placebo, and neither the participants nor the researchers typically know which group that they are in, and this means that it's double-blinded. So this design helps eliminate bias and keeps the groups comparable. Here, endpoints include firstly the prevention of the disease. So does the vaccine actually prevent infection with the target virus? Secondly, severe disease prevention. So even if mild cases occur and some people get infected, does the vaccine prevent hospitalizations or deaths? You probably heard of this a lot during the COVID-19 pandemic, that people were saying, I got the vaccine, but I still got sick. And yes, that can happen. But we were looking at the fact how less likely these people were to be hospitalized or actually lead to death. And we had, of course, have the long-term safety. So do we have any serious adverse events over time? Because we're conducting this study over several years sometimes. Of course, with the COVID-19 vaccine, it was done in a shorter time because it was a global pandemic, but otherwise it usually takes a longer period of time. And because these are large definitive trials, then the statistical methods are also more complex and rigorous. We also look at risk reduction percentages. So we calculate risk reduction, often reported as vaccine efficacy. You might have heard of that term also in the COVID-19 news. And here an example is if 1% of vaccinated participants get infected versus 5% of placebo participants, then it would be 80% vaccine efficacy, because you calculate it as 1 minus the risk in the vaccine group divided by the risk in the placebo group times 100. And you will also be looking at the Kaplan-Meier curves that we discussed in experimental studies. So we're not looking at just people getting infected or not, but we're asking how long do they stay free from infection. On the y-axis of this curve, you would see the percentage of participants who remain infection-free. And the x-axis is the time since vaccination. And this is why you can observe that if it drops, that there are people at a certain time starting to get infected. And this way you can visually compare vaccine and placebo groups over time. If the vaccine is effective, the curve for the vaccine group stays higher for longer than for the placebo group. And of course, the Cox regression models that we also talked about would calculate adjusted hazard ratios. And the hazard ratio compares the risk of infection at any given time point between groups. And adjusted hazard ratios account for participant differences, again, talking about the confounding variables such as age, comorbidities, or exposure risk. So if you have an adjusted hazard ratio of 0.3, it would mean the vaccine group has a 70% lower risk of infection at any given time compared to the placebo group. And if you sometimes read published articles of randomized controlled trials, you might see intent to treat or per protocol analyses, which are analyses that check robustness to see both real-world effectiveness and ideal adherence effectiveness. So intent to treat analysis is where every participant is analyzed according to the group that they were assigned to, even if they didn't follow the protocol perfectly. And it actually reflects the real world effectiveness because, as you know, people sometimes miss doses or drop out of a certain treatment. And per protocol analysis is where only participants who follow the study rules are included in the analysis. So it shows this ideal adherence effectiveness and best case scenario results. So if you compare the intent to treat and per protocol analysis results, then it gives you a full picture of how the vaccine might perform, both ideally and also in the messy real world. And we have regulatory review and approval. Of course, this is a very important step in any clinical trial, not just in vaccine research. And regulators such as FDA or EMA in Europe would require full data sets, pre-specified endpoints, and sensitivity analysis. And they vary that findings are not just statistically significant, but are actually clinically meaningful. So we have to be looking at this real world. So our effect size is clinically meaningful, because if they're just statistically significant in a certain study, it doesn't mean anything. We need to prove that we have a clinically meaningful result that actually will help the global population. And after approval, monitoring still continues. So as I said, we have the phase 4 of clinical trials, even though this is already when the certain vaccine or drug is on the market. And here we have certain methods. Firstly, we have passive reporting systems. For example, we have a vaccine adverse event reporting system in the United States. Then we have active surveillance using linked health records to track outcomes. We have observational, cohort, or case control studies for new or rare side effects. And we can also have a cohort study, so follow a large group of vaccinated people and compare their health outcomes to a group of unvaccinated people over time. Or we can do a case control study where we start with people who developed a rare condition and look back to see whether vaccination rates were different in these people who developed the rare condition compared to controls. And here, statistical signal detection becomes more important. So you're looking for higher than expected rates. So if usually we would expect one case of a rare neurological disorder per 100,000 people per year, but after vaccination we see 5 cases per 100,000, then that's a signal that is worth investigating further. And adjusting for confounding factors such as age, gender, or health conditions here is also important. And as you know, vaccine research is built on core ethical principles. Firstly, we have respect for persons, which is that we collect informed consent and not just a signature. So we perform a whole informed consent procedure where the participant can ask questions and we disclose everything about the trial, every risk that we know, and we don't just make them sign a piece of paper saying they will participate just because we told them the benefits of it. Then, of course, we have beneficence, where we protect participants from unnecessary harm. And we have justice, where equitable access to trials and eventual vaccines for everybody. And especially when research happens in vulnerable populations, then ethics must be central. In this case, it could be in elderly people or in pregnant women. These are all vulnerable populations. And of course, as in any research, there are also challenges in vaccine research. Firstly, we have to balance speed with thoroughness, for example, during pandemics. And this is why a lot of people were worried in the pandemic about the speed at which the trials were happening. We have to recruit diverse populations for generalizable data, but this might often be hard. And we have to navigate public trust, particularly when misinformation spreads, as you also saw with COVID-19, especially through social media and, let's say, blog posts instead of actual news or actual research articles, that a lot of people were focusing on the wrong type of media for disinformation, and that spread misinformation. And in conclusion, firstly, robust research methodologies and statistical methods are essential for producing credible science. Vaccines exemplify how research principles must be carefully applied, starting from the lab to real-world application. And as future researchers, clinicians, or policymakers, you must be critical consumers of research. So you must ask not just what were the results, but how were those results obtained, and from there establish whether the study was robust enough, whether it can be trusted. And that will be all from my side from the theory today.